first study we're doing here is in Huntington's disease patients in the early stages of the disease. So stage 1, so the early side of the disease. We're not targeting juvenile Huntington. We're not targeting the children or people up to age 25, because that is a little bit different in terms of its severity and pathology. But the typical Huntington's disease over age 25, people who are in the early stages, so they may have just learned they're diagnosed, but people who are known to have the SNP. And we also, we screen when a patient comes into the study, they're screened for the SNP to make sure they have it. And then they're entered into the study, and then the drug is administered intrathecally. So it's through a lumbar puncture. And we administer the drug, and then a month and thereafter later, we actually measure spinal fluid to see if we're able to reduce mutant Huntington. And the ability to do this, to measure this biomarker, the mutant Huntington biomarker, really has been one of the things that has advanced companies wanting to sort of go into this space. You know, my previous experiences in multiple sclerosis, where that was most of my drug development experience in MS, most of my academic experiences in multiple sclerosis. But one of the things that really revolutionized drug development in multiple sclerosis was the availability of a biomarker. So in that case, it was MRI lesions of the brain, brain scans. And that ability to detect the effect of new drugs completely changed how drug development happened in multiple sclerosis. And now there's numerous drugs for multiple sclerosis. And the way it changed it is because in a relatively short period of time, you can take a group of patients with the disease, try a treatment, and then through something very non-invasive, just doing brain scans, see if the drug has a potential effect. And it allowed companies to reduce the risk of studying that disease. And when you reduce the risk for a biotech company to study the disease, they're more interested in doing it. And then that leads to innovation. That's where we are in Huntington's disease. You have a biomarker that finally you can measure that's directly related to the gene. And you can measure it by lumbar puncture, which is not non-invasive, but still is a routine procedure that neurologists do. And in a relatively short period of time, see if you're making a difference. That you've seen an explosion in interest in Huntington's disease, an explosion in interest, and similar story for ALS and frontotemporal dementia. That's really what made the difference. And our trial is designed to take advantage of all of those features and give us an answer, hopefully relatively soon, on whether we have the potential of a new treatment.